Good afternoon, everyone, and thank you for participating in the new webinar organized by Prodotti Gianni. As many of you already know, Prodotti Gianni always attentive to innovation in the field of life science, organize webinars for the presentation of products or technologies useful in scientific research. And today's webinar will focus on the presentation of multiplexing analysis that can be performed in flow cytometry in a simple and fast manner using RayPlex arrays. The webinar will be held by Dr. Jared Wilson, Associate Director and Business Development Scientist for RayBiotech. And you can put your question in the chat box at the end of presentation. Jared will be happy to answer. Moreover, you will find my contact details in the chat box, which you can use for any further information. And the webinar will be recorded, and you will have the opportunity to review it on the YouTube channel of Prodotti Gianni. Okay, thanks again for participating. And Jared, if you are ready, you can start.
Okay, can you hear me all right?
Yeah.
All right, good. If I'm looking over here, I'm looking at you all, and over here is my screen. So if you have any questions, punch them in the chat box. Maybe Roberta or anyone who's has audio can like cue me in. I'll try and pay attention to, but bring them up as they come, because I'm going to do a high-level overview of arrays in general, as well as the RayPlex technology, and then we'll dig in a little bit deeper. But if anything's not clear or you've got a fun technical question, just reach out and let me know. So again, I'm Jared Wilson. I'm Associate Director of Business Development here at RayBiotech. I also do a lot of high-level experimental design. I'm an immunologist by trade. I got my PhD at Emory and did a postdoc at Emory as well, and then followed that up. Really joined RayBiotech almost immediately out of my postdoc after 2 years. So I've been here a little over 10 years now, and it's been a fun ride. So let's see how this is going to work here. All right. Just a quick introduction of RayBiotech. If you are not familiar with us, this is most of our team actually in front of our building. We were founded on the backbone of the very first commercial planar antibody array. We have a headquarters here in Peachtree Corners, Georgia. It's just north of Atlanta, if you're familiar with the state of Georgia and the city of Atlanta in the States. We've got about 35,000 square feet here. I'm not sure what that works out to in meters, probably something divided by 9. And about 95 employees here working. There's about 25 of us in the front office, and the rest are in the lab side space. We're GMP and ISO certified. If this matters and you have questions about it, we also do GCLP certifications and work, and we have a CLIA lab as well, which is a more diagnostic approach. So just if you're not familiar with arrays and why you might want to consider them just from a high level, they're a very cost-effective way to kind of profile some sort of set of proteins and the expression pattern. It's a good way for either a screening tool or, as my boss likes to mention, as a hypothesis generating tool. You've got a drug or a treatment on some cells or in some mice, or in a patient that has an effect, but you have no idea what's going on. There are lots of different ways to profile the proteome, be it mass spec or arrays, as I'll talk about. Mass spec is great if you don't know what you want, you don't know what you're looking for, you got to kind of scan for everything. Arrays are great if you can narrow it down a little bit to focus in on a target, or as well as we've gotten better at arrays, even now at doing an 8,000 plex array to kind of give you just anything and everything, and that's kind of our goal. Arrays also help you eliminate some of the post-detection analysis steps, because you don't have to do any data analysis. We've told you what the target is. You're evaluating the target. It's very sensitive to some clinical very sensitive to some clinically relevant samples as well and targets. And there's no sample depletion required, which is often an element of mass spec that you may not appreciate. So lastly, just the pros and the cons of arrays. So I view the pros as the targets you're probing. This, I think, is both a pro and a con, but a very big pro in part of it, because you don't have to do the back end analysis to figure out what you looked at. It's very sensitive to lower expressed targets, which can get into some limitations in mass spec depending on how good your sample prep is. Arrays are much faster. They are faster in turnaround time, as well as you can run a lot of them at the same time. And again, you don't need to pre-purify or pre-prep your sample. On the con side, again, you know your targets. You have limited your field of scope a little bit. That is a bit of a con for arrays. You're not scanning for everything yet. It's less sensitive to highly abundant targets. Arrays, you've pulled in a bunch that you want to run at roughly the same dilution. Some of the higher end targets fall off. And then again, arrays right now are manual or semi-automated. But all of this really speaks to some of the complements that you have with arrays. You know, mass spec usually needs help on the lower end, depending on how good you are at sample prep. That's what you have arrays for. You did your mass spec array or your large target array proteome screen. You want to narrow it down. Where do you go from here? You don't need to go back to mass spec. You could do an antibody array as an alternative, be it custom or pathway generated. And then again, as if you needed pathways, you know, post-translational stuff that mass spec isn't super good at, you could use arrays for this approach instead. So some Ray Biotech arrays, I just want to kind of highlight that we are developing or have developed, and really the idea is that we could just focus on anything and everything specific to specific pathway designs, diseases, molecular types. I've given some examples here. I'm not going to cover them all. Just I wanted to give you an idea of kind of some that we do focus on. And then also our arrays are highly focused as well on non-human, non-mouse, non-rat species arrays as well. We're starting to develop these. We've had some great collaborations with some agricultural groups as well as some aquariums even that have some things they want to look at and are looking for a tool that they can bring on board in their labs. So the two real types of antibody arrays, as I think of them, there's some in-betweens here, but largely these are the two examples. You have a planar surface-based array where you've got a glass membrane or glass or membrane support that you've printed your capture antibodies onto. That will serve as the surface that everything is on. You'll run your samples on the glass slide. They're individual wells, or you've created wells. And then you do all your detection on top of that, and you read that with a microarray reader or a chemi reader like you would a Western. And then there's the suspension-based, flow-based arrays, like cytometric bead arrays or Luminex is the most popular. We went ahead, so we've been doing the planar arrays for forever, but every time we talk with customers, we would get the question of, oh, we want Luminex, right? And well, maybe not everyone. A lot of people are very familiar with Luminex. Luminex is, of course, the technology underlying a lot of the Plex offerings from certain companies. They are licensing in the technology using their pairs. We wanted to kind of go away from that because with a Luminex system, you need a dedicated machine. And a lot of companies that are interested in this already have a flow cytometer. And so Ben on our team took a look at our antibody pairs. Ben's our flow cytometry specialist. He took a look at our flow pairs and wondered if he could just use kind of the cytometric bead array approach and make his own antibody arrays using the same pairs and doing a multiplex and single plexes and things. And that's really how RayPlex was born from a suspension side. As I mentioned, these are our three. I just want to briefly kind of show them. You have your membrane, your glass, and your RayPlex array that we're really using by just any standard cytometer. It's designed specifically around, all you need is a red and blue laser. When I was in grad school, a long time ago, I won't say how long, we had a fax caliber that had enough lasers to run our RayPlex arrays, and really that's how they're designed. We can make them easier to apply to, I could say, every flow cytometer. I would, but I'm pretty sure every flow cytometer on the market and in a lab that's functional right now can run our arrays with both the red and blue laser. And that's really where it's focused specifically is, you don't need a specialized cytometer, you don't need a cytop or some really high level cytometer. You can use a standard basal unit. So the RayPlex arrays are designed, again, around the cytometric bead array approach. We have individual populations of paramagnetic beads. Well, they can be paramagnetic. They're not always. I'll get into that. The beads have several different sizes, which allow you to sort them by side scatter, if you're familiar with flow cytometry. So side scatter takes a look at, it shoots literally across the side, and the bigger the particle is, the more it scatters, so side scatter. And so bigger particles appear higher in the spread, and then we have a large, a medium, and a small type bead. Each of these beads also has a little bit more or less fluorophore inside them. What I'll be showing here is A647, which is your blue laser channel. So We'll be looking at that bead population by that. And then that'll allow you to discriminate, this bead is coated with, let's say, TNF alpha antibody. It's a large bead. It's a large bead in a large population, and it's got the lowest fluorophore of 647 in it. So you can gate on that. If you're familiar with that, you can group the population and focus only on that bead. And then we'll take a look at how much PE fluorescence is on that, which is indicative of the target. Because the bead has a capture antibody on it, it will grab the analyte in the sample, wash it off. We'll come back with a biotin-related detection antibody. We use biotin to increase the signal and not just a directly PE labeled antibody. I can show you some data on that if you're interested offline, but the biotin works better at amplifying the signal. So we've got a detection antibody that comes in and also grabs the target that's bound to our bead. And then we come back with strep PE to grab all of the free biotin. And again, the streptavidin and biotin interaction increases and amplifies the signal of the PE because more can bind to the tetramer of biotin. And then we look for that PE signal as a measure of how much target analyte was bound on the assay. So we take a look, just to introduce the bead populations, so you kind of know what they look like. If you are familiar with flow cytometry, scatter plots, this will be very familiar. If not, I'll kind of try and walk you through a little bit of it. There are, I think y'all can see my mouse according to how this works. There are three populations. There's this large side scatter, 8 micron size. There's a medium one at 5 micron, and there's a 3 one. We name each of these individually. You'll see down here we have B1, B2, B3. Up here they're unlabeled, but just they're colorized just for for visual effect. Each bead population, 3 sizes. We also have different populations of fluorophore labeled to that. So just internal to the bead, it has more or less fluorophore inside it. This is on the manufacturing side from the bead manufacturer we work with that does this. So this is a bit of a proprietary mix, but they juice some beads up a little more than others, and you get these nice populations of 647 in the small beads, right? So each one is a different bead, and each bead is labeled to a different specific antibody. So I'll get into some examples here, but like let's say this is TNF alpha and IL-2 and interferon gamma. We can say these are all the interferon gamma beads, and we can gate this one specifically with this square or with other techniques and look at the amount of PE fluorescence presence in that for how much target it's bound. We're looking at some other bead colors as well. We wanted to limit it for now to 647 because again, we want it to apply to all cytometers. But we have had some customers wonder if they could do more multiplexes or more larger panels of a multiplex by using different beads. So we have identified some other bead options as well to expand it further. But those will probably be limited to commercial arrays for now, just because customizing them is a bit tricky. But we are able to customize the standard bead set for now. I'll talk about that in a minute. Okay, so once we've gated on our bead here, I've gated on just B25. I don't know what specific bead this is. We can look at the individual beads in this population. This is a histogram plot just overlaid across each. This is a standard curve, actually, if you're interested. So this yellow is the negative, and then each one of these is a 3x concentration of additional target analyte. So you can see this is the highest concentration, 3x dilute. It dilutes nicely on the histogram, and then next population. And this allows you to generate a standard curve with the amount of PE that's bound to the antibody. The more PE bound, the higher the concentration of the analyte that's bound to the captured bead. By generating a full standard curve, we can get a mean fluorescence intensity of each of these populations. So we might say this orange population has an intensity of 10 to the 4th. This is a mean fluorescence of 10 to the 4th. We can use that to plot it just like you would any standard ELISA or any other standard curve across this 8-point curve. And that allows you to fully quantify your eventual data against the standard curve that you ran in the assay. We have a great walkthrough of not only setting up your cytometer for the assay on the backside and on the front side, but also on helping you analyze the data. We can even generate a free analysis tool to be included with the RayPlex. We have one ready, but it depends a little bit on your analysis settings. So if you're using FACSDiva, the tool is ready. If you're using FlowJo, it takes a little bit of a different tool to do the data analysis because FlowJo exports into a different format than Diva. I'm not sure what everyone's using, but we can try and make something work for you if you need an analysis tool to generate the data and help with it. But we have a great walkthrough in the manual of kind of how to set up your cytometer, get it ready, as well as how to analyze the data. And so just some quick perks. Again, we're not requiring a Luminex system. If you're familiar with Luminex, you're familiar with flow cytometry, you can run this array. It's very straightforward. The 4-hour assay with about 1 to 1.5 hours of hands-on time, depending how good you are, how automated your system is. Again, it should be compatible with pretty much any cytometer. Use about 25 microliters of sample. This is like a dilute ready to go sample. You could probably dilute down depending on your targets, but that's your kind of sample amount. Commercial wise, we're going to have as high as 30 target arrays available. Our highest is 25 right now, but again, we did develop that additional colored bead set for specific cases, so we'll be able to expand that. We're right now looking at the 5 to 25 range. For custom arrays, though, probably going to max that out at about 15, just because it's unlikely you're going to give us some targets that don't interact with each other. And a lot of times for customs, we want to have a good bead spread, so you don't wonder if bead 1 population is overlaid with bead 2 population. Just cleanliness and to make sure the data looks good. We see some great CV between the runs, because we're measuring so many beads. We can tell you how many to capture. It's at least 10,000 per population, but you can capture even more depending on how many beads you flow through the cytometer. With 5 to 10% CV, it's quite good. It's fully quantitative, as I mentioned. Picogram per mil sensitivity. It's very cost effective as well. Once you get over 4 targets, it's usually cheaper to do a multiplex, regardless of the multiplex. RayPlex is included here. Than to do an ELISA. It saves on sample as well. Other reasons to consider it is the savings on sample. I can't stress enough. Like for some sample types, CSF, depending on the animal model you're working with or how young the model is, even serum plasma can be short on volume. It's a really good one to consider. And another perk, if you'll work with us on any RayPlex purchase, we will validate your sample type. So if you want to call us or email us and say, hey, I've got, I don't know, rat urine for my assay. I want to run this rat RayPlex. Can you make sure rat urine works with your assay? We will either work with you to get some of the example sample, or we can go buy from a vendor, rat urine, validate the assay on it, make sure that it works, or let you know that, maybe one target is going to be less sensitive because of the sample type. Would you like to proceed? We'll do that. It's a real good perk by Ben's team. He's really trying to make this as best as possible for the end user. So if you have some questions about a sample type, let me know. Let us know. We'll validate against it. So data analysis side, just if you want to see what the end-line data looks like, this is a lot of numbers, and I apologize, but this is the rat inflammation array 1. You can see each population of beads has its label against it. So taxon 1, gamma interferon. I hope you can see this. We're also labeled them both in the manual, on the website, in the software, about what population. Red 1 is the highest bead at the lowest 647 setting, and it Z stacks if you're interested. Red 1 is interferon gamma. And then if we run these standard curves, we can see that this is what the concentration should be, and this is the signal that we get out of this is the signal that we get out, and we can see the standard MFI drops from standard 1. Standard 1 is about 15,000. Standard 2 drops by a little bit, 8,700, and it drops all the way to our blank signal. I think even this one has some nice wiggle room between the lowest setting and the negative. And then we plot this out just like you would any other ELISA. You can use a 4-fit parameter curve, which works best, just to account for any sort of S shape in the curve. Also, a lot of these will be quite linear through the middle. So if that's where your samples all exist, you could just use a linear curve adaptation. And then if you're interested, the LODs here for just this array in particular are on the left. We calculate these. You can also calculate these individual per run based on the interactions between the lowest standards and the negatives. I can give you some math for that if you'd like, but we found that each lab has their own preferences on LODs and things. So happy to advise. As I mentioned, again, a free analysis tool is available. If you order this, let us know. We'll give you one. We will make it work for whatever your analysis tool is or your analysis software is. I'm not sure what everyone is using in your. I only used FlowJo forever until I got to my postdoc where we started using FaxDiva a little bit just because it was pre-installed on the cytometer. So I'm only familiar with those two, but I can work you through others as well. And Ben's got more experience than I do, so he and his team can help. So some array options that we have available, just if you're interested. This isn't all of them, but this is a good kind of snapshot of ones. So right now it is focused heavily on human, mouse, and rat. Our first kind of soft launch of this was some human arrays that went really well, taught us a little bit too. And then since then, we've been launching these a little bit more. Again, I mentioned we can fully customize some of these. We have full access to the entire Ray Biotech antibody pair list. So that's 3,500. Oh, yeah, you got it. So if you're interested in customizing, that's something we can absolutely look at and help you with. Again, you're limited to less than 15. We can even do a 1-plex if you really want to. We've had some people want that because of sample volume considerations, or they just want to use their high throughput sampler. So just let us know on that. But these are some example arrays, again, highly focused on cancer, inflammation, and growth factors, because these are not only the ones that sell the most, these are the ones that are most representative of our listing of products. We have also had some publications. So again, I mentioned we soft launched this, I think, in 2019. Had some good experiences with a couple customers. They end up publishing, and then more recently we launched it in full around 2021. I think COVID was officially over by then. You can see, so had some nice publications on some nice topics with an interesting selection of sample types too. It wasn't just one. I remember some of these cases directly when we kind of worked with them on the sample type. So I helped Ben out significantly with the flow cytometry aspects on marketing and technical support side. So I'm familiar with a lot of the flow cases. And really, that's it. I wanted to give a high level overview. Not a lot of science and technicality, but that's also why I figured I'd open it up for questions. If you had questions on the technicalities, or if I've talked too fast through a certain element, I can follow up there. If you don't want to put it in chat, I understand. That's my email address. You can email me directly. I'd happy to work with you, answer any questions, help you design something, tell you what's a good idea, what might be a bad idea, go from there. And so with that, I will open it up for questions.